Okay, so our last portion of the qualitative or the carbohydrate lecture concerns the qualitative test that these carbohydrates can undergo. And again, this is a lab that you're going to be doing in the class, and you're going to be taking some carbohydrates and you're going to be undergoing these qualitative analysis tests to determine what type of carbohydrate do you have. Do you have a 6 carbon carbohydrate, or do you have a 5 carbon carbohydrate? Do you have an aldose? Do you have a ketose? All of those questions can be answered by using these qualitative tests for the carbohydrates themselves. So we need to take a look at some of these tests. We need to kind of figure out what goes on, why do they do what they do, but none of the reactions, at least, will be on the test itself, but the information on the qualitative scheme will be on the test and presented. So let's take a look at first, what really are we talking about here. First, we need to talk about the types of qualitative tests that we have, and they're broken down into 3 major classes. The first class is the dehydrating reactions, and then the second class are the condensation reactions, and then in the third class we have basically copper reactions. So these are the 3 types of reactions that we're going to be dealing with qualitative analysis and with your laboratory that you'll do. The test reagents for the dehydrating acids, well, dehydrating means remove, hydrate, water. So it's removing water. And what happens here is that we create this structure called a furfural, or a 5-hydroxymethylfurfural. So I take a look at the al ending, and I probably think of aldehyde group. We'll see if that's true. I see the furfur, so this is similar to maybe furanose. Maybe. What was furanose? It was a 5-membered ring. So maybe there's a 5-membered ring in there. We'll see when we look at the structure. But, hydroxy in the front, well, hydroxy looks like OH group, so one of these has an OH groups, and the other one didn't mention anything about hydroxy, right? So furfural, or 5-hydroxymethyl furfural. So we'll take a look at that structure here in the next few seconds, but we can at least break down the name and maybe kind of predict, hopefully if we're lucky, what this thing should probably look like. The presence of furfurals can be detected by adding a condensation reagent. Okay, so that we'll talk about in a minute. The next one up is this condensation reagent. So phenolic compounds that react with furfural, or 5-hydroxymethylfurfural. So phenolic is phenol. Phenol is benzene ring, aromatic ring, something of that nature is going to happen here with the condensation reactions. They're highly colored products, so these colors are very, very pretty when you take a look at them and you do this qualitative scheme. Don't be surprised if you see these beautiful greens and beautiful blues and these vibrant reds, because you will see them. These tests, 3 of them that you will be doing here in this class, will be the Molish, the bowels, and the oneoff. So these are the 3 tests, or 3 of many that you will do. I'm just listing 3 of them here. And then finally we've got this whole section of copper ions. So we're looking at what happens as far as copper is concerned. And copper, we're looking at specifically copper 2, and we're looking at what happens in the reaction itself. So copper 2 can be reduced down to copper 1, or vice versa. So reagents that contain copper 2 ions can oxidize carbohydrates. And there's 2 of these tests, Barfoids and Benedicts. Both of these have copper compounds. Copper traditionally is blue in solution, and the reagents for Barfoids and Benedicts don't be surprised if you pick that reagent bottle up and you see a blue color because copper is involved. So sugars that react with copper 2 are called reducing sugars. Remember, we talked about this whole concept of reducing and non-reducing sugars, and this is where it comes into play. Okay, so let's take a look at furfural. Here is furfural. So Al ending, aldehyde. We see the aldehyde group here on the end. This C double bond O group with an H that's sandwiched out here. It's not drawn, it's understood to be there though. And then we see furfural, which is fur, which is 5-membered ring. So 1, 2, 3, 4, and 5-membered ring. So we kind of predicted it, but that's the structure for furfural. There's many sources for furfural. They're listed here on the slide for you. They smell like almonds most of the time. So when you pick up these compounds, these pure furfural compounds, they do have a nutty odor to them. They smell quite nice actually for an organic. So if I take a look at the first organic qualitative test for carbohydrates, we're going to first look at the Molish test. And Molish will specifically test for a carbohydrate. It will let you know, yes, this is a carbohydrate, or no, it's not a carbohydrate. So that's basically what the Molish will do. Now, the Molish will consist of using sulfuric acid. So it dehydrates the carbohydrate, and it removes the water by using sulfuric acid to do so. And then this forms a product. And we take that dehydrated product and we react it with phenol. Now there's a couple of different phenols that we can use. Naphthol, alpha-naphthol, resorcinol, or thymol are both okay. And those structures are here for you. So this is the alpha-naphthol, this is the resorcinol, this is the thymol. So these are the three structures that we're after. Those are all phenol sources, right? There's rings in each one of these. The reaction of those two reagents, if I take that phenol, react it with the dehydrated product, it gives me a nice red or purple color as far as the test tube goes. So the sample's mixed with Molish, sulfuric acid is slowly added down the side of the tube, and 2 layers will form. At the interface, a positive test means purple ring. That is what you're looking for. So you don't mix these two things together. You take the sugar with the Molish, you add sulfuric acid slowly down the test tube, you'll see 2 layers that will form. At the interface of those 2 layers will be a purple ring if it's a carbohydrate. So that's the positive reaction for a carbohydrate. Okay, so let's take a look at this reaction. This is the reaction of glucose with sulfuric acid to make this 5-hydroxymethylfurfural, and then this reaction is going to be reacted with a phenolic compound such as alpha-naphthol. We get this structure that we see here in the center, and this structure basically goes down to a purple colored dye, and that is what we see in these test tubes when we do the Molish reagent. Molish, the reaction, as far as the reaction goes, this is something that you might want to put in your lab book, but it's not something that I would necessarily study or learn for the test. I will at least tell you that I will ask, what is the purpose of the Molish? So Molish qualitative test tells you if it's a carbohydrate or not, period. So that will be a question, but I won't ask about the reaction, what kind of compound is formed, or what's the structure of the product that gets formed. That is something that I will not ask, but I do want you to have a general knowledge of what this reagent does. So with the Molish, what we're going to see, and there's the mechanism for you if you want to take a look at it. But with the molish, this is the concept of the positive test. This is what you're after. So again, the molish reagent mixes with the sugar, and then that gets put into a test tube. Sulfuric acid is slowly dripped down the side of the test tube, and what you will see here at this interface is this nice purple color. So this purple color that you're seeing here is the positive identification for the molish. If that purple color shows up, then yes, you have a carbohydrate. Now it's your job to figure out which carbohydrate you have. If the purple color does not show up, then that means you probably don't have the carbohydrate in this case. And if you don't have the carbohydrate, you don't have to do any of the other tests that follow it. So you label it as no, it is not a carbohydrate, and you move on to your next sample. But that is the positive identification for the molish. Beautiful purple at the interface layer. So you're not looking for a solution that turns purple, but that interface, that immiscibility line, that is what will turn purple when it happens, if it happens. The next test that we can talk about is something called the bowels test, and the bowels test is very specific. It looks for pentoses, so it's looking for 5 carbon sugars. So this is a way that we can determine if we do have a sugar. Do we have a 5 carbon sugar? Well, we can use bowels test to tell us yes or no. Yes, you have a 5 carbon sugar, or no, you don't have a 5 carbon sugar. So this is broken down into 3 pieces: orsenol, hydrochloric acid, and ferric chloride. So if the sample is a pentose, you will change it to a furfural again in this reaction. And once the furfural is formed, you then react that with iron 3 chloride, and 2 things can happen. It can turn blue or it can turn green. Now, you've got to be careful, because the green color, depending on the sugar, well, the green color could be green, or it could be a brownish green, or it could be like a red brown. So we see kind of an array of different colors that can happen there. But if it turns blue, and that's really the only color that you're looking at, is the blue color. If it's blue, then this is the positive test for a pentose. If it's green, red, or brown, or any mix of all of the above, then that is the positive test for a hexose. So this is how we distinguish between a 5 carbon and a 6 carbon sugar. So the recipe for the reagents there, you can kind of see hydrochloric acid is used in it, ferric chloride is used in it, orsen's used in it. The reaction requires a little bit of heat, so these test tubes that you put these samples in, they have to go into a hot water bath for a couple of minutes. The reactions are going to be there for you if you want to take a look at them. Here is basically the picture of what the bowels test will look like. So over to the right hand side, that is the blue color that you're after. So that blue color means that this sugar compound that was tested was a pentose. It's a 5 carbon sugar. If you take a look at the other 2, these are like greens and browns and maybe a little reddish hue in it. Well, these are 6 carbons. These are hexoses that the bowel is letting you know what you've tested for has 6 carbons in it, not 5 carbons. So we can begin to whittle down the number of choices, and that's what this qualitative test is all about. We use molish to determine, is it a carbohydrate, yes or no. We then use the bowels test to determine how many carbons do we have. Do we have 5 carbons or do we have 6 carbons? And this will let us know. Now we've narrowed our choices down even further. The next test that we can mention is something called the oneoff test. And the oneoff test distinguishes between aldoses and ketoses now. So once you figure out how many carbons are involved in the sugar, you then need to know, do I need to look at the aldose chart or do I need to look at the ketose chart in order to figure out what sugar I'm trying to ID? Well, the oneoff will do that for you. The oneoff is very similar. You take the sugar compound, you're going to add some reagent to it, typically resorcinol and hydrochloric acid, very similar to the bowels reagent. You put it in a hot water bath, you heat those things up, And depending on the speed at which it changes, that will kind of tell you whether an aldose or a ketose is involved. When heated, the ketoses are actually formed quicker. The products of these ketoses are formed quicker than the aldoses. So it's all about time here. Time is very important. In those lab books, you should be writing down the time that these reagents changed with the Seliwanoff test, because that will tell you whether it's an aldose or ketose. The positive test here is a very deep red color, cherry red. Sometimes you might go pinkish, but a lot of times you're looking for this cherry red color that will show up in the actual test tube. And of course, if these things are heated and we time them, a hot water bath is going to be required. So there's the reaction for the Seliwanoff test, and here is the positive ID for the Seliwanoff test. So you're taking a look, this is the end product, it's this brick red color, and you're really wanting to know how long it takes for that brick red color to show up. Typically, all of the sugars will go this brick red at some point in time. 5 and 10 minutes, those are typically the cutoff times, and that's what you'll see basically in the lab write-up itself. It will tell you measured at 5 minutes, did it change? If not, continue to heat it, and then continue to heat it until you get a 10-minute time. Did it change then? If not, then stop it, and just label everything else from that point on. So that's the positive ID for Seliwanoff. You're looking for a red color. Benedicts. Let's take a look at Benedicts. Benedicts is basically used to detect the presence of aldehydes. Ketoses are detected because they are converted into aldoses using a base, right? We've talked about that reaction. That's why that reaction was important. If I put a ketose in solution, that ketose can give me epimers, right? Or that ketose could turn around and give me a aldose as well. And this was our very first reaction that we talked about in this lecture series. Okay, so if I take a look at these ketoses and the Benedicts test, well, Benedicts is used to detect the presence of reducing sugars. So it doesn't really distinguish between aldoses or ketoses. Since ketoses do show a positive test like aldoses do, what it does is that it will tell you if it's a reducing sugar or not. And we've defined what a reducing sugar was. It's this anomeric carbon that has this free OH group. And if it has that free OH group there, then it can begin to react with other things, and we call that a reducing sugar. So typically monosaccharides versus disaccharides, that's kind of what we're looking at here, because disaccharides tend to form this glycosidic bond that covers up that free OH group. Not every time, but traditionally that is the trend that people see. So that's why the last statement is there. Benedict's test for all monosaccharides and the disaccharides lactose and maltose. Lactose and maltose do have a free OH group that are out there, and that can participate in these reactions. So we do see a positive ID for lactose and maltose because of that. Benedict's reagent, typically it's a mixture of sodium carb, sodium citrate, and copper 2 sulfate. You're looking at copper ions, so the solution's typically blue. When it reduces down to a copper 1, a precipitation reaction happens, and you get this copper 1 red brick color precipitate that forms in the water. It also requires a hot water bath. So this is kind of a before and after picture. What you're seeing on the left is the Benedict's reagent mixed with the sugar, so you see kind of a pale blue color. Then when you put that into a hot water bath, and then after 5 or 10 minutes you remove it from the hot water bath, if there's a sugar involved that has a free OH group, then you know that this is going to turn a brickish red color or pinkish red color, and a precipitate forms. So this is what you're seeing here. That's why it kind of looks a little cloudy on the screen, because this is a precipitate red and not a solution red. So that's the difference there. You see some particulates that form in the solution itself because you're forming copper 1 oxides, and this is a precipitate that's swimming around in solution. So that's the positive ID for Benedicts. If you get a positive ID for a Benedicts, then you know that you're working with a monosaccharide, basically. If you don't get a positive ID for a Benedicts, then you're working with some type of disaccharide or polymer. And this is where Benedicts really comes into power. Right. The next one is Barfowoods, and if I take a look at the Barfowoods test, well, this helps detect the presence of monosaccharides as well, and this also uses a copper solution. This is a mixture of copper 2 acetate and copper 1 oxide. Copper 1 oxide, of course, forms this precipitate again. Disaccharides react as well in this case, but they react much slower. So when you do the Barfowoods test, immediate reactions typically give a very good signal that it is true, in fact, a monosaccharide. A disaccharide, though, takes a little time, so they will eventually react just like before, but these disaccharides will lag in time. So you're looking at 10 minutes, 15 minutes, or maybe even longer. The Barfowoods reagent, copper acetate, and 1% acetic acid. So again, none of these are really nasty. This is just a copper solution with a little bit of vinegar mixed in. Vinegar is actually probably a little stronger. Okay, so here's Barfowoods reaction if you want to take a look at that reaction. And this is really a true copper precipitate that you see here. This is very distinct. You do see that copper precipitate fall into the bottom, and then you're left over with this copper extra that's still in solution, which is why that's still a bluish in hue color. So this is the positive ID for a Barfowood. So if you do have a positive ID for Barfowood, it is a monosaccharide. Maybe 10 or 15 minutes down the road in a hot water bath, if you don't see this precipitate form, then you can assume that true, it is in fact a disaccharide or maybe a polymer that we've given you instead of a monosaccharide. The Tollens reagent we've talked about before. I'm not going to really spend too much time on this as far as the qualitative test goes, but the Tollens reagent we said was very explosive. The Tollens reagent should be diluted after you use it or after the lab is finished with it. If not, it makes this sodium azide, and the sodium azide is what they pump into your airbags in your car. So that is what makes this thing explosive. You kind of want to stay away from it if it's made fresh, and if it's been out for, weeks or months at a time, this thing can build up in sodium azide, and this can make it go pop. So there's the reaction with the Tollens reagent, and there's the beautiful kind of test that the Tollens will give you. So if you're doing the Tollens reagent in the lab, you'll take a sugar solution, you'll add Tollens reagent to it, and you'll just shake it, shake it, shake it, shake it, shake it. And then when you do, the silver begins to precipitate out if there's a reducing sugar there. And when the silver begins to precipitate out, what you will see is that the silver will latch onto the sides of the glassware, and you'll get this mirror image that forms this silver precipitate all the way around the outside of the glass. So that's the positive ID for the Tollens reagent. So that's the story with the qualitative test. I hope you have fun with them, and hopefully you can pick out your carbohydrate that we've given you. So we don't pull any tricks, nothing up our sleeve, but it's there.